Okay, our final session for today is lab notebooks. It's actually being presented by one of my graduate students who I know collects lots of data and does a very good job with his lab notebooks. So I think we can all probably learn from him. But his name is Dhanush or Dan Amarasekara. And so he will talk to us a little bit about lab notebooks, hopefully a pretty light session, and then we'll adjourn for the day. So I'll turn it over to you, Dan. Thank you.
Thank you, Dr. Fitzkee. So in the session number 10, I will be talking about the basic rules you have to follow to maintain a good lab notebook. So in this session, I think most of you are already started working in labs. So for some of you, it will be a review. But if you are new to the research environment, I think there's a lot to take away from this session. So these are the major focuses in my talk today. What is a lab notebook, and what to write, what to include in a lab notebook, and different ways that you can use to maintain it in a good way. And throughout the presentation, I will show you some examples too. So what is a lab notebook? It's a primary record of research. Think like it also reflects what you are seriously, how serious you are on your research. So your peers, your PI, or someone else, if they ask how did you get this result? Can you prove it the way that you did? So you should have enough information in your lab notebook to show it. So if someone else, if they want to follow or to reproduce the results, so you should have enough information there to show, to guide them throughout the process. So that's the major purpose of a lab notebook. So whenever you started working on the lab notebook, so if you have a physical one, so the first couple of pages, that is for the table of content. So if you don't have that is for the standard way that a lab notebook is. If you are not using a standard one, then you can leave the first couple of pages just to use it as the table of content. So why do you think the table of content is very important? So the major two things I can come with, first thing, if someone asks, hey Dan, do you know how to prepare this protein? So I don't need to go through all the pages to find it out. So I can easily, if I have it in the table of content, I can say, oh, here it is. It's easy. And let's think I made a protein last year, and I want to do it in this year. So it's a long time. I don't know the things that I did last year. So I can easily go through the protocol, and I don't need to document it again because I did all things in that year. So it's not wasting the time that you have. Then the next part is whenever you start something in a day, I normally do it I use a new page and I put the type of experiment that I'm going to do. I put the date. Then next part is the experimental details. The procedure, as I explained, it should be clear in writing so someone can follow. Then the list of regions. Why the list of regions is important? You are using a certain chemical for your research, and you have it from different manufacturers. So if you know in some chemical it had an impurity, if you're going to use it in your experiment, that's going to be bad, right? So you should know which chemical you use for your experiment, that then in the next time whenever you use it, then it will give you the same results. Otherwise, it is from a different manufacturer. Sometimes a certain impurity in that might cause a problem. So the best thing is whenever you document everything, you should have the manufacturer's name and also the catalog number of that region. That's better. The next part of the experimental detail is the list of instruments. So the major thing I can thought of in this part is if you have only one instrument, that's fine, that you have to use it every time. But think about you have multiple instruments. For example, you have 3 different UVs in the lab, and you are using think that they are labeled in 1, 2, and 3. Then on a day, like you are using the second instrument to take a measurement. But after like couple of days that you get to know like, oh, on that day that it was broken. Oh, that you use the third instrument. So it was not broken. So you know like on that day, like I used this instrument, so it was working fine, so my results are good. And this instrument that was bad, so I have to repeat that experiment on the other day, right? So that's also a good thing to list the instrument that you are using whenever you are documenting things in your lab notebook. Then after like collecting the data, you can find a way to like present it in a manner like using a graph or figure or gel. Then with all the data that whenever you can come to conclusion from this experiment that I did, that I can come to this conclusion, and you can write it down and you can document it in the lab notebook. So I think this is one of the most important slide in this session. Like so what to document in the lab notebook? Is it like only the good results that you're going to put in the document in the lab notebook, or the good and bad results? What do you think about that? So it's already there. Like, so think like for most of the researchers, they don't get the good results in the first place, right? So they will do several trials, then they will change several parameters, then they will go to the good results. So most of, like for me, my experiments, I was like struggling for like 6 months to get good results. So which means if I didn't document it, which means when I will never get the good results, because I know from the bad results, you can have some experience, right? So why it is bad, you can get to know it, and you can change parameters, then you will, like stepwise, you can go to good results. So it's better to get, to document all the things like good and bad results. Then the next thing, like honesty, like whenever you get the good results, is it good to like remove the pages from the bad and throw it away, so you have the good, right? Then you can remove it. So then if someone says like, oh, Dan, you're getting all the good results. Wow, you're a good scientist. So it's not that way. Like you should be honest. Like it's not good to remove any pages from the lab notebook. And like no need to skip pages. Like if you skip pages, you can like cross it out. Or if there's any unused parts that you can cross it. So from this slide, so all the things that you are doing in the lab, you have to document it, even the results are good or bad. Then you should be very honest. Okay, so I need your help with these two panels. Like, what do you think why the top panel is bad and the bottom panel is good? Okay, you can't read the mistake. Why the mistake is, okay, that's correct. So why do you think like it's a mistake? So it's like totally black.
You can't read like the
yeah.
mistake on the top.
Okay, so, okay. So what?
You want to be able to read it so we all make it again.
I didn't get it.
I said you want to be able to read it so you don't make that same mistake again. That's why.
Okay, that's also good. So what do you think, like, it's bad that because it's making a mess? What about that? Like, lab notebook is a mess. Is it? I didn't get it. Date? No, there's no connection with the date. No. So what do you think, like, the person who thinks it's 0.531 is correct and 0.325 is wrong, right? That's why he has, like, that's his mistake, and he thinks like 0.531 is correct, right? At that instance, like he got to know, then he like cut it because he thinks like whenever like he after a couple of days or couple of months he get know like, oh no, it's 0.531 is wrong, 0.325 is correct. So what will happen at that point, right? So the first person, he has to do the experiment again, right? The second person, he already has that. He can still read his mistake, but Actually, at that time he thought it was a mistake, but actually it's not. Okay, so the second person wins. Okay. Okay, then whenever you try to document things, it's better to use a pen. Why is pencil is not good for this purpose, for documenting? Why is that? Yeah, I didn't get it. No, you can easily erase it, right? Yeah. So it's better to use a blue or black color pen, and make sure like whenever you are, if you are working in an organic lab, so if it's exposed to most of the solvents, it's not going to like fade away. So that's one thing that you should consider, and only use the ruled areas for documenting, and you don't use any unnumbered pages. Whenever like possible, like always like use supporting data. You have to attach the supporting data with all of your, whenever you are documenting things. So in this one, like it's like a simple test that you can do. Like so as I told you, like you can use a pen, and I told like it can, whenever it exposed to some organic solvents, it can ruin, and you will like go away with your data. So which means like they have done a simple test like this. What you can do is you can take a paper and write with different pens, and you can expose to different organic solvent to find out which is the stability of your ink, like is it like bleeding or not. So this one, like the journal references. So whenever we are doing the experiments in our lab, we can't come with our own protocols, right? We are taking protocols from different people. So that's coming from the references. So think that whenever after you working, like you totally forgot about this one, and after a couple of months, whenever you come to submit your work, then you find out like I am missing where I got this protocol. So this is the best way to, this is one of the way to document the journal reference. So the first author, year, journal, volume, and first page is all things are there, so you can easily find it out through the internet or using the Google Scholar, and you can put it in as a reference. Okay, this one, how to do calculations how to document the calculations in your lab notebook. So here, what I'm showing is the major thing. Like think that you're preparing a sample and it's not working in your way, like in the way that you're thinking of. So you can find out whether it's a problem with the instrument that gave the result, or it can be like whenever you're preparing the sample that you made a mistake. So from this that you get to know, like because we have all the calculations, if you've done something wrong here, that's why it didn't give you the expected results. So that's, the calculations are very important to document in the lab notebook. So this is another calculation. Okay, from this part, who really owns the lab notebook? So it's like, because all of us are coming from academic environment, so all are working under PI or an academic advisor. So which means he or she is the one who owns the lab notebook. So it should be always stayed in the lab, and whenever you can ask for the permission, like if you want to get photocopies or like you can take the pictures, that you can ask the permission. And it's better like not, if it is a physical one, so like I also encourage you, if you like you can have like a digital one of your own, like then you can no need to take pictures and so you have a digital one with you. In this slide, I'm going to talk about handling complex digital data. Anyone like here like doing NMR? So whenever you do NMR, like it's making a lot of data, right? In the first place, it's making like a lot of data. Then you have to go through different types of processing, and you will come to a figure like this at the end. So this figure, it's a 2D NMR figure, like it's in one dimension it is 15N, the other dimension is proton, 1H. So whenever you come to this type of, like, whenever you make this type of figure, like it's a higher, like there's a bigger deviation from the original data. That means actually that if you ever want to do any changes to this figure, if you want to do different processing for this figure, you have to again start from the original data. So it's the major thing that which emphasized from this slide is like always you have to keep in mind that where's the original data. You have to store it in a secure place, and also you have to back up your data. So here in the next slide, what I'm showing is what it really shows is a data that is related to a chromatogram. So this is like a protein prep. So in this figure, like he or she has mentioned like from where the original data is stored. So you can see like March 15, 2010, size exclusion dot dat. That's where the file is, and it is in the Smith data directory. And all the details about this chromatogram is there in page number 65. And whenever he done his protein prep or his or her protein prep, like he or she has used the fractions from C1 to C5 for further study. That's all the information are there, so you can easily go through it whenever you need it. Like most of people, they are looking for the original data whenever it is near to their submission. So it's better to have it, know the place, and have it in a specific place that is secure. So I like to give you a couple of minutes to think and discuss, like, in what ways do you think, like, do you benefit by keeping a good lab notebook? Any answers?
So you can remember and keep track of what you've done, especially if you're doing like the same thing for like a couple of months or even like a year, just so you can have a log of everything you've already done.
Yeah, that's a good answer. Any other answers? Okay.
Really, so that when you go to publish a paper, you can look back on what you did, no matter if it was like 2 or 3 years ago and you hardly remember.
Thank you. So in what ways do others benefit?
I know for my group, I work under a grad student, so if I keep a good notebook, she will be able to see what I've done and use it in her final dissertation or research.
And if others want to replicate your research or your methods, it's more easier for them to do so.
I didn't get it. Thank you. So we got some good answers, like it benefits us and also benefits your peers, right? And also the PI. So with all of that, I like to say like the current policy of our lab. So whenever we join the lab, one of the thing that we had to do is we had to like purchase a Dropbox account. And whenever we do our experiments, all the data, all the protocols, all the things are going to our Dropbox account. So whenever it is close to the submission, what we are doing is we are having a shared directory. Again, it is in Dropbox, and we are putting all the things into that Dropbox account. Then earlier we had all the data and all the protocols with us. Then we are sharing it among us, so all of us have the access to it, even Dr. Fitzkee. Then in that shared directory, we have like subdirectories, like for manuscript, figures, EndNote library that Joanna will be talking about it tomorrow, and then original data directory. So with all of that, we have all the things in a certain place, so it's easy to handle, and whenever it's for the submission, it's easy to work with that. And most of the journals, sometimes they require raw data to be available for publication. So we don't need to worry about that whenever we try to do the submission, like we are ready with everything. And again, like why why Dropbox? There are a couple of things like to mention, like Dropbox is secure and it will help to back up all our data. Whenever we do something in our laptop or in the personal computer, it automatically syncs it, and we don't need to worry about that. And if think like your computer is broken, like we don't need to worry, like you can access it from a different computer. And there's another thing that our lab does is we have a common lab server. So all the things that in the shared folders it goes to a different like computer. So whenever someone leaves, that data it's not going to disappear, like it's still there. So after this, let's go and take a look at some examples. And I think you can download it through the bootcamp website. Okay, this is Dr. Fitzkee's. Yeah. So this is like example from Dr. Fitzkee's. So he has put the date, April 29, 2017, and this is like the heading inoculating pin 1 construct. So I think you got to know like about pin 1 a lot from the PDB like he did last time. It's also pin 1. So what he's going to do is he's going to make the pin 1 protein by using cells in the LB medium. So in the first part, what he's showing, like whenever, I think you all are aware of how to make proteins, right? So first part is to inoculate the cells in a small media and then move it to then inoculate in a large media. So that's in the first part what he's showing, the OD values in this part in this table. So then he has all the things what he has done, the protocol, and he has one result here like running a gel to find out to see whether the protein he's interested in is the pin 1 is there in the cells. So from that, what is he's showing in here is like he got, so you can see this is a pretty close to the expected value. So from the gel that he's coming to a conclusion. And another day, May 1st, 2017, then he's going through some, I think he's going to do FPLC and SCC. So for that, he needs some buffers. So he's showing how to make the buffers. And these are the sequence of the proteins. Okay, so this part is important. Like, so this is FPLC results of the pin 1 protein, and here you can see where the mouse cursor is. Like, or it shows where the file is, the original file is stored. So that's important. So like again, this one is like the final part. I think it's related to the SEC, like size exclusion chromatography. So that's why it is showing 2 peaks. So again, like in our systems that we normally we are having like an account for different people. So you can see like he has worked under, like there was a student called Dhanush. So her account, so all the things related to the result file is there, and all things are documented in a proper way. So from all of these, that you know, like what are the things that you have to do whenever you are working in the lab. So especially whenever it's something which is related to your research, what are the things that you have to document, and whenever you come to a conclusion that it's better to write it there. And if you are missing something, if you if you got some unexpected results, you have to write that one too. That I didn't get these results, and you can write it down like why I didn't get that result there. So from all of this session, those are the things that you can take away, and that's all I have. Yeah. Thank you.